So welcome everyone. Good morning, good afternoon, and good evening. Our speaker, our AI influencer has joined us. I will introduce Dr. Shamir Khader, PhD and MPH, Global Head of Computational Biology at Sanofi, has 15 years experience of interdisciplinary scientific and leadership experience focused on optimizing drug development using data and AI, leading global teams in data science, AI, genetics, and bioinformatics, has expertise in clinical, preclinical, and post-clinical applications, developing a comprehensive disease research data ecosystem at Sanofi, and has experience working in and with AstraZeneca, Mayo Clinic, Mount Sinai, Northwell Health, and Philips. Holds a PhD in computational biology and MPH from Imperial College in London. Has over 150 peer-reviewed publications, conference abstracts, and patents. Today's topic: from task-based models to foundation models in medicine, emerging trends of AI in medicine. Please, Dr. Shamir Khader.
Thank you, David. Hello, everybody. A very good morning or good evening or afternoon, depending on where you're joining from. It's a pleasure to be here, and I'm really excited to have this interesting sort of meeting. I'm looking forward to the dialogue part, so I'll go through my keynote, and then looking forward to have an interesting discussion today. So what I want to do, I hope you can see my right screen. Is everything good from your side?
Yes, it is.
Brilliant. So what we're going to do is, AI is on its own, as you all know. We've been through this whole ChatGPT moment. Where is all of this going, and why are we here? This is one of those questions. And what I wanted to do today is to kind of give you an ultra short course on, where we are and where things could go, with a few examples. Hopefully we can cover all of them, or at least some of them we can do. I've been a student of AI, and I continue to practice AI in different aspects of life science, medicine, and pharmaceutical development. And then I also continue to be a student because AI never stopped improving. So the idea really here is today to tell you what is happening in this space, right? Like, where are we going with respect to modeling? What happened? And let's say if you are interested to bring in AI to your practice, or if you're interested to leverage AI, what type of approach you should be potentially using in. So that's what really I wanted to do with the roughly 18 minutes I have. There is a huge data boom in medicine. We are collecting more data than ever before. That's one of the things. But at the same time, when we have been using this data, there was a whole big data wave in medicine, and then now that wave is now merged with the AI wave as well. But then what happened was that everything about AI changed in between. So the idea really is to kind of connect these two, or rather three trends about data, about AI, and about computing together, and see how this entire thing will change in medicine. And thank you, David, for that kind introduction. And my research interest, really is about looking for signals or looking for patterns across different types of data. This could be data that comes from traditional clinical landscape. It could be a simple blood test that you do, or I hope you can see my pointer, or an EKG value, or you do an OCT of a patient, of a cardiac reading, or things like that. Or it could be pretty much conventional molecular omics profiling. We can look into genomic data, transcriptomic data, proteomic data, metabolomic data, microbiome, and then also a little bit of unconventional. For example, exposome, is just about the clinical and biological. No, right? Like, we are also, we interact a lot with our community, with our environment. That also plays a significant role in our health and state of health as well. But in the end, can we use all of this data to automate, predict, classify, sense, respond, and act, so that we can have better outcome in health. So really, we do generate a lot of data, in my opinion, but then is it the right data? Is it the app data to solve the problems we have? That's the kind of things that I get excited about. I'm not going to go through this because I hope some of you have heard about this, but what I wanted to share is that there are different ways people define these terms, whether it's big data, predictive analytics, data science, AI. We will be covering some of that. But then again, what happened is that there are sort of rather, as we can think about, so far we have three sort of school of AI. So what you call as an all code AI, where you build a solution all by yourself. Hybrid AI. This is basically in terms of you basically have some form of human input into your AI solution, and then sort of no code AI, fully automated. This is where we are right now, what we call as agentic AI as well. And these are driven primarily, in terms of, all code AI. What I meant by that is, let's say you want to, you want a machine to read an EKG and give you an output. That's one of that example. Maybe it'll read that and tell you that, okay, this EKG include, or this echo is for a hypertrophic cardiomyopathy patient. And then the other one is human in the loop, is gives you some signals but not give you a conclusionary one, but you take that decision with the AI as well. And then finally, it not only takes the decision, it will also generate the report for you, pull in the additional results, as well as why it thinks. It kind of give you a little bit of an explanatory aspect as well. So with that, what we're going to do is, in terms of AI, why AI, and what is really AI, and why is it different from very classical paradigm in computing? A simple definition would be this one. It's one of my favorite way to kind of explain that. In traditional programming, you have a computer, you bring in data to that, and then you bring in a program, and then you go through the data, you get an output. So basically, it's a systematic or a sequential approach. That's what you do in a traditional programming. But in terms of machine learning, what do you do is you take the computer, you'll bring it, you'll show them the data, you'll show them, okay, these are 100 cat pictures, these are 100 dog pictures, and give me a program from that. And that program is what I will then use to do the classification further down the lane as well. So that's what we are. And then we talk about, artificial intelligence. There are different types of, approaches there. Within that, one of the field is machine learning. And then another emerging field was deep learning. Now we are all in a version of, or rather an extension of deep learning called reinforcement learning, is where a lot of development actually have happened. And why all of this happened? All of this happened because we have been riding this data wave for quite some time. Whether you take, biology, or you take chemistry, or you take financial. So we are generating, collecting, and managing a lot of data. But then collecting data doesn't solve any problem. You need to have right infrastructure, right type of questions you need to ask to this data, and that could ultimately help you. And we know this has been happening for some time. But especially in biomedicine, what happened is that the collection of the data was like, as you know, that the cost of sequencing a genome, it's been told that it will come down up to $100. Give or take, we have that right now, but then there are some nuances to that. But we continue to generate a lot of data, whether it's genomic data, whether it's protein structure, protein modeling data, which recently won a Nobel Prize for AlphaFold. Or looking into other types of omic data, right? Like, for example, interaction data, protein interaction data, how DNA interacts with protein, how drug and proteins interact, how different genes in the setting of different diseases interact. So a lot of data, a lot of knowledge we have been building. And then we are now at a convergence point where we have a lot more data that is not amenable to traditional statistical approach. You definitely need some AI and machine learning. So you have, on one side, you have all the data and other approaches, and then on the other side, you have this.
I think we lost you, Dr. Khader. We have some technical difficulties. Wait one moment.
Yeah.
Yes, you're back now.
Okay. So what I'm saying that, we have different approaches in machine learning. You can, I'm sure this has been covered, but you can do unsupervised, supervised, or reinforcement learning. Supervised is when you know the label of your data. Unsupervised is you want to explore trends within your data, and reinforcement is like the one I mentioned, where you use a human in the loop or other types of agent or a policy within your machine learning approaches. So these are sort of the rather three major approaches. So we've seen the growth in data, the growth in AI approaches as well. But then along with that, that's not, and what happens with this connection between, biology, chemistry, pharmacology, and drug discovery. Ultimately, a biological idea will be converted into a therapy, and then that will be delivered in healthcare. But here, there is a whole sort of a paradigm shift happening. So we are moving from a traditional data types to evolving data type. Data can be anywhere. So let's say you have 10 hospitals in your network. Earlier, when I used to work for large academic medical centers in the US, we used to bring everything in one place. But now we have tech to do that. You can do federated learning now. We can always handle large amount of data. We used to handle petabytes, but now you can handle exabytes easily as well. Structured or semi-structured, unstructured, doesn't matter. Data model can be evolving. Data can be, very high dimensional. You can, of course, use statistical approaches, but along with the statistical approaches, you can also use machine learning or deep learning approaches as well. Cohort size, you can handle large number of patients as well. You can, I'm a big fan of both hypothesis and data-driven approaches. That's also another way that things are evolving as well. And along with that, so think about this whole aspect where we are capturing a whole lot of data, comes from a different approach. It could be genetics, could be the environment, it could be the clinical readout, could be the disease progression rate. But then again, along with, we started gathering a lot of this data, but then along with that, we also looking at different aspects of AI. For example, one example I can tell you is that if you are in the US, you might have seen, or in other parts of the world as well. Tesla and Tesla's, autopilot is one example. Like, different companies have tried that before, but they optimize that, for example. And again, that's one example of how you literally bring in, visual AI in one way. So it can not only drive, it can see, it can brake in advance, and it can do so many things together. So that.
Oh, sorry, we lost connection again, Dr. Khader. We'll wait one moment. There. Okay, I think you're back now.
All right. I'm sorry why this is
Oh, no,
happening.
no worries.
Okay, we'll continue. All right. So we saw all of this. You have data, but then you also have algorithms. But then again, you need computing, right? Like, so as a physician, and most of the physicians in the call, that there is a lot of data we capture when a patient is in the hospital. Like, you have your nursing flow sheets, you have data that you capture in your notes, you have the data in your hospital's electronic health record, you can have data coming from your sensors and wearables. But how do you bring all of this together? So this is again the last piece. Like, you need a robust computing infrastructure also to really do that. So to kind of come to that first part, so you need three things. You need data, you need computing, and algorithms. So this is why we were primarily in the space of AI. So this is how we got here. And again, you can, you take any branch of medicine, you can apply different types of algorithms. So in this case, you wanted to apply a
Excuse
simple
me, Dr. Khader, I think we lost your slides.
oh,
Yes, we only have your cursor.
let me join back
Actually, I
again.
thought it
Maybe.
was fuzzy logic AI for a second. They
Yeah,
had
let me join back again. Maybe it will come back.
okay.
Yeah.
Yeah, for a second, I thought it could be the AI trying to figure out what the cursor was. So, Dr. Kadir will rejoin us here in one moment. coming back in now. Okay, welcome back. Yes, now we can see your slides. And I think you are on mute.
Yeah, can you see
There
now?
we go.
So
Yes.
this is this is actually how if you are interested in designing a machine learning project, this is how you go about it. You define the goal of the project, you acquire the data, you do the cleaning and processing, you explore the data, is the data right one, you do some visualization, then you go into a series of activity where split your data into training, testing, validation. You either do feature selection or feature engineering, select your appropriate algorithm, do model tuning, generalize it, and then testing, evaluation. Finally, you will have an interpretation and the report. That report will be, you want to make sure that it's insightful and it describes what the model is. And depending on the country you are from, you probably have to file for regulatory submissions, and then again make sure that it's reproducible. So this is how we were doing. This is pretty much the same even now. I will share this slides as well, but then this is at the core of if you have one problem, whether it's you want to predict patient outcome, you want to predict for example, you want to predict uh, patients readmission rate, or you want to predict how likely a patient to come back for the next patient visit as well. So any kind of use case you can build using a framework like this. So I'm not going to go deep into each of this stuff. So what I'll do is I'm going to skip a few slides in the interest of time, where you can do, look, browse through the slides. And this was written for an invited paper for American College of Cardiology. You can see that there are some really nice tips that if you are interested in building a machine learning model for healthcare. So this is all well and good, right? So that's sort of the first era of machine learning. And then everything changed because you had what you call as graph-based machine learning coming in. What happened is that the traditional prediction model, you were primarily dealing with data in array format. And then things change. So in here, what you have is you have hypertension and different medicines for hypertension. And then that different medicines have potential application in other indicators. So very classic repositioning example. But then what happened was that we can now represent data in what you call as a graph format. And each of the node or the edges can be loaded with different types of information, actually. So that came into picture. And this kind of changed a little bit of the approaches that we've been doing. So till now, I've been talking primarily about machine learning using array-based data. Think about it, data you see in an Excel file. But here I'm talking about data that is in graph format. And we spend a lot of time about this approach, and because this was driven by number one, graph theory, and then also based on network science as well, which has a lot of interesting way to mine through large amount of networks, so large amount of data as well. And we've been using this technology and applying it, to try to understand how you can use clinical trial data. Can you use this data to develop, therapies for not just an indication disease, but a combination of indication disease and its comorbidity? Because a lot of times, patients move from the classical definition of diseases and move there. We've been using this to find new targets. We've been using this type of approach to find, not only find them computationally, but also validate them in diseases, including cancer. So these are all of them are published work, so you can definitely refer to them, how we have applied this. So that this is what I think, from a task-based models. These are still task-based model because this will only do one model at a time, and then we have been slowly shifting towards foundation models. And this is where what we see that so far till 2017, more or less, AI was like this, up to graphs were there, but then everything changed. And everything changed because starting from 2014, things have been shifting towards neural networks, pre-training, fine-tuning, transfer learning, and emerging approaches like, beyond training, testing, and validation. We have been looking into attention networks. We have been looking into embedding the data. We have been looking at encoding the data. We've been looking at approaches like transformers. And then, thanks to this huge amount of work, again, this is also one of the things that won Nobel Prize this year. So about the neural networks, there are different types of neural networks. You have encoders, you have attention networks, you have generative AI, and starting from simple perceptron models. So how do you mine through these type of complex data using these approaches? So that has become very easy because you could do GPU computing, you could bring in all the data as well, and we have been applying this to a variety of problems. And here is one example where we use neural network technology to see whether you can predict, uh, early indicators of breast cancer from mammography data. So we build a whole algorithm around this approach as well. And then again, when you come to pre-training, what is pre-training is it refers to the initial training of a model on a large diverse set of to learn general representation of the input data. What I meant by that is so let's say 5 years ago, if I want to create a model for doing breast cancer latent expression from mammogram, I would rather have to start collecting hundreds of thousands of mammograms and then do that. But these days, what happens is that you could get a model that's already built and trained using that data. And then let's say you wanted to look at a specific aspect of breast cancer in mammogram, you can then pre-train that your model with that data. You can fine-tune that model with that data. And here is where the foundation model actually comes in. So you can bring you can use an existing model, and then you can use that existing model as a potentially as a foundation model, which is and then refine it further. You can pre-train it or you can do fine-tune it for the specific task that you have. So what happens is that model that's trained with tens of thousands of breast cancer mammograms is your foundation model. You can then bring different tasks to that. You can then tune it actually. So this is where we are. This is how, if I am to do the same use case of going, looking for potentially early markers of breast cancer in mammograms, this is how I will do this time. And then again, you can move things from, transfer learning is again another technique where you can move knowledge gained from one task and you can apply it to another one. So let's say we're looking for latent expression, but you can also look for delayed expression as well. So that or late expression of cancer as well from that. So that's something that you can do with transfer learning. So we've talked about pre-training, we talked about fine-tuning, we talked about a foundation model. Then transfer learning is again another concept. So you can move things from, let's say you have a lot of data on one disease, whereas you don't have a lot of data on another disease, sort of related disease, so that you can do some transfer learning as well. So what we do really is you leverage pre-trained models that have been trained on large data set for a particular task, and then you reuse that and adapt them for a different but a related task as well. And then the other important approach is embedding. Embedding is you take large amount of data. I showed you a network of drugs and diseases, but then each of as a graph, but each node can be then loaded with load of other information. What is that drug? What is its chemical type? What are its side effects? You can go to the target, and what is that target? What is it? What is its biology? So you can embed a lot of different types of information. So this is like you can embed in the level of words. You can embed sentence. You can do graph embedding, knowledge graph embedding, and whatnot. So that's again another concept. And then you can also do something called encoding. And I might ask you one example. If there is a name called Bensi, what would come to your mind? Um, Maybe name of a girl or something like that, and then if I give you a little bit more information, it's a 1-year-old, it's from MD, like to play with water. So I'm giving you a little bit more information. And then if I also tell you that Benzi it's a puppy, then what happens is that I encoded all of this information, or your brain encoded all of this information, including the picture, all this information in here. So this way you can actually build a lot more, data into your models. This is also another aspect that's possible. And then this is probably one of the most important piece of work. So we've been able to develop what we call as attention networks, and an attention in AI, and this is what we call as the uh, one approach actually that is an important aspect of how our brain works. So let's say you are going somewhere this on this fine Saturday morning, somewhere right after this talk. What would be in your mind when you're on the road? You might be looking at the traffic. You might be looking at the speed. You might be looking for if the scenery is good, the weather is good, you'll be looking at that. But irrespective of everything else you do, your attention is always on the road if you're driving or if you're walking. You'll be looking always on the road. So that is called the attentive mechanism or attention mechanism. And then this is there in human cognition, where we prioritize certain information or others. So what attention networks, what these computation models that we build now have, they can be attentive to something. So you wanted your model to look for specific patterns. You wanted your model to learn specific aspect from the data. They can actually do that. So attention in AI refers to a mechanism that enables models to focus on specific part of input data. And this is where why foundation models work, or why multi task models work, because it can do this. It's a critical piece, critical work. It's not an easy paper to read, but it's one of the most important work in our era in machine learning. And then with all of that has come into picture, now we are at what we call as transformers. So transformers refers to a neural network architecture again, and this is primarily applied in a facet of AI called natural language processing to mine text data. And then this is basically built to look for sequential data efficiently. So where are you? Then where are you driving, where are you going, where are you we don't say where are you swimming, where are you smiling. So the smiling is not the least probability, right? Like probably driving and going and will be the highest, and then comes swimming. So the idea really is, if given certain type of information, can you predict the next sequential word, or sequential, it could be other types of data as well. So this works because it also use the attention mechanism we talked about. It works because it can also be trained using large amount of data. It works because it can be also built using the approaches like foundation models as well. And they have remarkable success. And everything that you see in GPT, the GPT stands for generative pre-trained transformers. They are transformers which are based on attention network, which have encoding, embedding in the past. So what I wanted to try really was to show you this is where the field has gone, from traditional, Excel based or array based data to graphs to this type of large scale models actually. So this is where we are, and of course everybody knows about GPT as well. And then this is another sort of example you might have seen. You can take these sources and these destination and build these type of faces, and none of them really exist because these are all imagined by AI, for that matter. And then large language models are transformers, and then this is how different companies have been working on this. And this is nothing new. Like I said, after 2014 these things happened. Google had a lead, but then eventually came OpenAI and other approaches. And then they typically run a benchmark, and this is what you typically get for each of these different types of model, whether you're a BERT fan, GPT fan, or Llama. This is how you benchmark them. But they're not easy to build. Like it's not like something that you can build, because there is a significant computing requirement needed for really good models. And then this is just the cost. It's a little bit older, but I just want to share that. And then this is a model that I created yesterday, or a table that I created yesterday, just just to show you how much it costs to build some of the medical models as well, right? Like BioBot was 500,000, clinical bot again, but MedPalm or MedFlamingo or Radiology Llama 2, so it's in tens of millions of dollars, whereas an NIH grant is very very small compared to all of this. So these are very very expensive. That's where you need to think about pre-trained models, using them in and build on top of these. They'll be much better models that you will get. And and then again, this is what's happening. So we saw most of these steps within a GPT. You have a transformer encoder, you have a pre-training, we have a decoder, we have an attention mechanism, personal encoding, and fine-tuning. So remember that, where are you going example I've been telling as well. Uh, did I lost?
I think we're about at, we let you go, run over a little bit,
Okay.
about extra 10 minutes here because we have one of our Dr. Innovators cannot make it today. Would you like to just summarize, Dr. Khader? Then we'll move
Yeah, I
on.
can do that.
Okay,
So there are a few
thank you.
examples here. I would, again, you can apply AI across different approaches. So here is one where we applied this to, potentially look at patients who are admitting to an ICU. And here is another one, how you can use AI to do potentially assess patients who have a likelihood of readmission. And then again, large scale cohorting using a foundation model as well. I have three examples. And before I close, these are all published data. None of these are from my current employer. These are all published, and these are all the work is available in the public domain. And this keynote was more of a pedagogical exercise for me, and I'm happy to take any questions online or offline, and looking forward to continue to engage and have some downstream discussion. All right.
Oh, thank
Thank you.
you.
Back to
Thank
you,
you.
David.
That was absolutely fascinating, Dr. Khader. And we will continue to share, would continue to uh, share some of the things that maybe you didn't have a chance to get to. We're going to start with our Dr. Innovators, and then any of the participants in the audience that have questions after our Dr. Innovators, please feel free to go ahead and put those in the chat now. I wanted to introduce Dr. Sean C. Jones. He's going to do his best to make it. He is performing service in Kenya, Africa. He's there quite often. He's been going for many, many years, and he's going to do his best to get here. So he may join us later. I'll let you go ahead and read his bio summary here. And I'm going to ask you to go ahead, in the interest of time, to read the wonderful backgrounds of our Dr. Innovators as they ask their questions. Please, Dr. Hafsa Nasir. Dr. Nasir, are you somewhere where you can unmute? I believe. Yes. Okay. Thank you.
Good evening from my side, sir. It was an excellent presentation from Dr. Shamir, and so many technical details he has given us. It's really fascinating that AI is advancing so much that we living in the developing countries are quite fascinating for us. So I would like to know some from Dr. Shamir, some practical examples. The Sanofi Aventis, I think from his experience at Sanofi, what type of um, genetic data they have used in any of the drug discovery or drug repurposing or for drug tailoring, like personalized medication. Some practical example uh, he can explain it.
Thank you, Dr. Hafsa Nasir. Thank you for the questions, and great questions. So, pharmaceutical companies, it's been sometimes since that pharmaceutical companies have now started investing in this area. So you've asked about a few different things, and I can tell you, we build some, from with respect to genetic data. Sanofi has something called Sanofi Genetic Data Lake, where we compile data from a variety of genomic data sources that's internally generated, coming from our collaborator, coming from public consortiums, coming from CROs, and that's one way. So we then use this genomic information to look for potentially targets, but look for signs for safety, look for new mechanism of actions, you look for other types of connections between the genotype and phenotype. So that's one aspect that we do. And then you also asked about an important part called patient stratification. So, given that, a particular medicine have an efficiency in a give or take 30% of the patients, you know, is there a way to find them? Is there a way, if a company have more than one therapy for one indication, can we, personalize them? Can we stratify patients so we can target them with utmost efficacy? That's one of the dream, and we have been, companies have been doing this in the oncology space, but then the other disease areas still an up and coming, and then of course Sanofi is working on a lot of different types of diseases under the broad umbrella of immunoscience. We have projects underway in inflammatory bowel disease, in asthma, COPD, whatnot. So different types of immune indications. But again, that's the path forward, right? Like the personalized medicine is where things will go forward. But really, the challenge is the machine learning we use, because if you have one disease and you have three medications available, how do you find the match between a medicine and a patient? So basically, you need to collect large amount of data on a cohort where you have responder, non-responder data, and then you build a model like the way we have seen today. You build a model using the approaches, one of the approaches we have seen, and then use that model before, as potentially as a companion diagnostics. You run a small test and then you do the matching. So that's the path really, but we're still getting there as we speak for many diseases. And there is also a cost aspect to that, and sometimes patients may not want to do a test. They want to get to the medicine faster. So there is all sort of that type of challenges there that we all have to come together to solve, because it's an open challenge, but we will hopefully we'll get there for sure. Yeah.
Thank you, Dr. Nasir. Do you have a second question or follow-up question?
Sir, I have a follow-up question regarding the genetic information we will need for this drug repurposing and drug trailering medication. What sort of, or I should say, how we are going to collect this genetic data? While living in Pakistan, I would like to know what is the simplest method we can utilize to collect this genetic data as and make a database of our country, let's say. How we collect and how it is processed further.
Yeah, so there are two things here, right? Like, so earlier, starting from the 80s itself, we were thinking, the Coumadin example, or, some of these classical examples where we were looking for, okay, you have this genetic variant, this is how you're going to, you are going to be a fast metabolizer for this drug or a slow metabolizer for this drug. So that was sort of the classical genetics approach we've been using in pharmacogenomics. But then starting from 2005 or so, we started to do, instead of doing genotyping, we were started to do, again, instead of doing, candidate SNP typing, we were trying to do genotyping. Like you can do genome-wide assessment. Nowadays you can do what we call as whole genome sequencing. You can do exome sequencing. You can do but only like 100 genes, thousands genes, depending on what you wanted to do, especially your use cases, that you can do that. And then a new concept came actually along with the pharmacogenomics approach. We now use something called polygenic risk score. So what happens was that when we did large-scale genomic studies, what we identified that there is no single, for most of the phenotypes, that single significant genetic variant was not really there, that we were all expecting that. But instead of that, what happened is that different variants of different types of effect size sprinkled across the genome. So, and even for drug response, it was like that. And then nowadays, everybody's using this concept called polygenic risk score to look for that. So that's one part. And then to do sequencing, what I would recommend, or in my experience in working with sort of large academic medical centers in the US, is start small. Start with a small cohort and start with the biobanking facility. What I meant by that is, this is what we've been doing at Mayo. This is what we have been doing at Mayo Clinic, Mount Sinai, and whatnot. A biobank is, you need to have access to patients' phenotype data, of course, by setting up a research program, getting an IRB, and following your local laws, you can have a research program to collect this. So, and start collecting patient data. Don't do, in my opinion, in the initial stage, if you have a specific medical specialty, you can collect patients in that. But, or you can get, population scale data, you can build a biobank. And then that can, sequencing costs are going down a lot, for example, and I'm sure there will be many companies would be interested to work with you and set things up actually. So that's one way to go about start small, start with the biobank. Start. You don't even need genome sequencing in the beginning. You may need only genotyping, which is again much cost effective than doing the whole genome sequencing and managing that data. Instead of that, you can do a genotyping as well. And speaking of there are significant amount of data contributed by Pakistani experts. I don't know whether you know about this project called Human Knockout Project. So this is actually a project that looked at patients, especially expat Pakistanis who live primarily in Britain, who have certain genetic variants that are detrimental to health, but irrespective of having that, they have a fairly long life. And what drives this? And this was one of the interesting thing that people have been looking at. That's one thing. And then again, a lot of this Pakistani population have contributed into Human Knockout Project as well. This is basically, you may have a loss of function genetic variant, and what happens, with some of them carrying that and they continue to live, some of them, have health issues as well. So how do you do that? So they already, in the global landscape, Pakistani population already contributing. So just wanted to kind of share that. Yeah.
Oh, that is really interesting and some very useful information. I've seen, for a number of years, organizations building, as you mentioned, the ethnic database is very important in Middle East, in Africa, and Southeast Asia. So let's move on, in the interest of time, to our second Dr. Innovator, Dr. Adebayo Moses Olugbami. I will let you read his illustrious career and background here as well as Dr. Moses asks his question. Please, Dr. Olugbami.
Thank you for having me. My question is with respect to how to use machine learning to approach early detection of pancreatic cancer, particularly with respect to identifying risk factors so as to be able to detect it early, either at the early stage or at before, or to identify patients at risk. So based on my experience here in Nigeria, I discovered recently that looking at the data of patients we have quite a number of patients coming from the eastern part of the country as our patients with pancreatic cancer. So I was thinking, would there be risk factors with respect to their environment or with respect to their tribe? So in what way can we use machine learning to look at data from this group of people with respect to pancreatic cancer, as to be able to identify risk factors among them and early detection of early stage of pancreatic cancer? I would like your input to that. Thank you.
Thank you, Doctor. Thank you, Dr. Olugbami. I hope I pronounced that right. So it's a great question, and one of the most important part. Can you preemptively, diagnose, right? Like, can you preemptively identify patients who are at higher risk? So in a world, we have all of our data in a massive health database, and we should be getting these type of insights way early, right? But that doesn't exist. And what you're saying is an important part. There are two ways I would go about, based on the data that you have, based on the data that you routinely collect, can you look for patterns there? So that's something I would do. I would look for, your regular blood work you do, or another other types of measurements that you do in your practice. Can they have any potential connections or correlations with pancreatic cancer? Is something I would do. That's one example. And then, the other approach, or sort of the approach that's kind of getting some traction here, is that doing some kind of sort of uh, liquid biopsy. What I meant by that is you can do a sort of a blood test that can potentially look for markers. This could be genetic markers. It could be markers of cell-free DNA. It could be markers of protein. It could be markers of metabolites. It could be a combination of this that you could potentially look for. And here in the US, it's approved for a few cancers. I don't think there is one for pancreatic cancer yet, but there is definitely one for broader cancers, but then sort of common cancers. But at the same time, there is one for colon cancer, for example. Instead of doing a colonoscopy, you can do this liquid biopsy test. So that might be the way to go. But then again, how machine learning would help, like just like we have seen today, if you have a set of data, set of patients, let's say 100 or 1,000 patients, you can look for patterns. If you know whether they end up getting the disease, yes, no, you can do a very classical supervised machine learning. Or if you don't, or you can also do unsupervised as well and look for patterns. And then somehow you can convert that into a simple test. I would say the success of a diagnostic test is not to build the most advanced or most complicated test, but rather the most simple one. And then, but at the same time, you can reproduce it across the world. And that's where the real value would come. That's where really this would go and become, sort of course, it should have evidence, but at the same time, this will be, important enough that it will change the way you deliver medicine, the way you practice medicine. So think about it in that level. If you have data, continue to collect data, continue to build data, and then also do biobanking of these patients. That's also important. And then another emerging approach that I see these days is like to predict disease trajectory. So if a patient have a certain types of disease, does that make him or her more likely to have another condition like Parkinson? This can be easily modeled from the data that you have. But you need the data. You need data for a few years, 1 or 2 years, and then you need to also have enough incident rate as well. Is it it's 1%, 10%, or whatever that is, good to have some incidence rate, and based on that you could build some conclusion as well. I wish you all the best with the goals, but again, a combination of analytics plus collecting the data, and then again sequencing could also help, but you need a multi-pronged approach. That's an important problem, very important problem, still an unsolved problem, but yeah.
Wonderful. Thank you. That is excellent suggestions, excellent feedback. Dr. Olugbami, do you have a second question or a follow-up question for Dr. Khader? If you could unmute and ask your question. I think you're on mute. If you can hear me, Dr. Olugbami. So we may have lost. Are you able?
No, hello.
Okay, there we go.
Hello,
Thank
I'm
you.
here. Thank you. My second question is about the choice of therapy with respect to malignancy still. Before we make choice of treatment, we will have done histology diagnosis, radiology, some molecular typing to be able to select the appropriate therapy for management of malignancy. I was going to ask about how AI and possibly machine learning can help with appropriate choice of therapy with patient with malignancy without necessarily going through all the stages of doing evaluation before you are able to prescribe treatment. Meaning that, is there a way we can have a faster, like quicker approach to selecting the appropriate therapy for patient with malignancy without waiting until you get the histologic diagnosis, molecular typing, and all that. Maybe that question became important to me because of some delays we have before the results of those investigations come out, which sometimes delay commencement of treatment for the patient. Maybe that may not generally apply everywhere, but if it is possible to be able to make diagnosis
That this is again another
early and to be able to select the appropriate therapy for patient with the help of AI, machine learning.
again, this is again,
Thank you.
yeah, an important and probably these questions defines, some of the challenges that some of the open challenges that we have in the field, right? Like, so we, given that in oncology, we do a lot of sequences, we do when a patient have, to look for the mutation, and based on that, we can definitely recommend some therapies. But then again, it has its own limitations again. So like what you're referring to is the real, oncology precision medicine that we are all thinking about. But again, AI can help. AI can look through the data, look through the responder, non-responder data, and come up with suggestions. But then again, here is where I wouldn't trust machine alone, right? Like I would rather take this approach of human in the loop, or a physician in the loop, or an oncologist in the loop approach, where we can help the oncologist to look through all the data, or look through all the past outcome data, and then he or she can then select the right therapy for the patient. That's one thing. The other important aspect is that, in terms of therapy matching, the challenge is that, especially when you talk about the timing aspects, it's important. And from my experience in working in oncology, the exposure actually, the outcome of certain therapies will depend on the exposure, whether you use this as the first line therapy or a second line therapy. So again, an oncologist cannot really make that choice without looking at some kind of a molecular readouts in the beginning. So this is where the challenge comes in. Good to have this molecular readout, good to have an oncologist in the loop, but then again, going faster is important because patient might be on a terminal stage. But then again, it really depends on various factors, including, whether this particular cancer have different subtypes have different therapies available in the market. So when you think about it, compared to last 10 years or so, we probably have now, a higher number of oncology drugs approved. So that's a good thing, actually. But is it all personalized? No, that's not the case. Is it all, in a way, matched to the molecular signatures? Some of them are, most of them are. But then again, we still need to continuously collect pre-exposure, post-exposure, and responder and non-responder data on these patients. And then also in oncology, the challenge is, whether the patient goes into to remission, and sometimes the tumor can come back in an aggressive form. All of that type of challenge still exists. So I hope I had a better answer, but then again, we really need to kind of not only build models for research purpose, but we need to bring them to patients, bring them through clinical trial, bring them through comparative effectiveness research, and then bring them to the clinic. That's the way to go forward. And that's actually taking time. AI is just one tool in that entire ecosystem or entire equation that you have there. But then it's really an important problem, and we should all think about it and collectively solve it. Yeah.
Excellent. Thank you very much. Excellent questions, excellent feedback, and suggestions and resources. Thank you. Let's move to our interactive Q&A session. We have a question from Dr. Ogundele in the chat. Thanks for an informative presentation. How can we, as Dr. Innovators, contribute local data from Nigeria and throughout Africa so that developed interventions can be effective in our population of patients? What are the processes or links to follow?
Excellent question. So one of the ways that you could, potentially contribute data is by joining research networks. And what is this research networks? Research networks could be, for example, there will be global networks for, we were discussing about pancreatic cancer. There are networks like that. That would be more than happy to take samples from the rest of the world, because a lot of times, a lot of the genomic or molecular data is really only available from European American or this part of the world. So definitely, companies and research organizations are looking to improve the research diversity. So that's one way you can be part of some of this research networks. There are, again, in terms of contributing to this approach, you can do what we call as, you can publish, your data as a paper, right? What I meant by that is, not every physician will have a large research team, but you can still publish this as a data. You can put that, put a data note into preprint servers. That's one way that you can, get collaborators reaching out to you. There are, once you de-identify this data can be contributed for research. For example, let's say you have access to genetic data. There are databases that accept genetic data with and without publications from around the world. You need to de-identify them. You need to follow your local regulations on, PHI and other approaches, and then you can use that as well. But the best approach would be to be part of a global research network, and depending on your area of interest, therapeutic area of interest, you will be able to find global research consortium that will be more than happy to have members from around the world, including Nigeria. Yeah, great question though. Yeah.
Yeah, excellent question, excellent feedback, and I've seen a number of now, of course, AI-powered medical writing platforms out there as well that make that part easier for you. Any other questions? Feel free to raise your hand, your virtual hand, or your hand, or just unmute. I leave it open so you can unmute and ask a question. However you feel most comfortable. You can type it in the chat or unmute to ask your question, or raise your hand. Any other questions? All right. Well, if there are no other questions, I think perhaps we will end there a couple minutes early, and we'll continue the discussion. If there are any other questions that you think of later that you would like to share, we will do our best to get you the answers. We look forward to seeing everyone back again December 7th, same time. Topic will be wearable and AI, real world patient data for better drug development. So look forward to seeing everyone. Here's an insightful dialogue. Here is a preview of webinar series 8 with Dr. Elias Zaid, Dominique Daniel Sofa, Dr. Innovator, Dr. Barbara Mikado, and Dr. Suleiman Tundi Stowe. So look forward to seeing you there. Have a wonderful rest of your morning, afternoon, evening. Thank you, Dr. Khader. Thank you, Dr. Innovators, and all our participants. See you next time.
Thank you.
Bye-bye.